Okay, so we'll start all over again. So today we're talking about corynebacterium species and bacillus species, which was a class meant for last week. And the outline which I'll follow is looking at or defining what the important biological or bacteriological properties of each organism, microorganism are, the epidemiology and transmission, pathogenesis and clinical findings, then go through the microbiological lab diagnosis, the treatment options in terms of mainly antimicrobial therapy, and then how we can control infection of the diseases. So starting off with the genus corynebacterium. So some of the species in this genus are part of the normal flora, mainly of the skin and the nose. And the microorganism tends to have some relation to the genus Actinomycetes and Mycobacterium. They are gram-positive rods and tend to be club-shaped, so similar to a baseball bat. So one end is tapering off, but the other end has a club shape. During cell division, they divide but tend to remain joined to each other or attached to each other, and therefore form what are known as either a Chinese letter or a palisade arrangement. The microorganisms are facultative anaerobes, they're non-motile, and they're non-sporing, and they're catalysts positive. So different species, the main one we'll focus on is corynebacterium diptheriae, which causes diphtheria. But there are other species that some of them, again, with change in epidemiology or interaction with the zoonotic environment that are getting transmitted to humans. So corynebacterium pseudodiphtheriae, which causes respiratory tract infection, but also endocarditis, wound infection, and infection of prosthesis. Corynebacterium ulcerans, which is mainly zoonotic, found in animals, but also transmitted to humans. Cirrhosis and pseudotuberculosis, which are found mainly causing in humans nasopharynx and skin infections. Their clinical significance is the importance in mainly individuals who have sites that could allow them to cause opportunistic infections. So individuals who are immunocompromised, individuals who have prosthetic devices in different sites of the body, and also in patients who are long-term hospitalized or in long-term nursing homes. In terms of history, so this goes all the way from 1613 all the way to 2019. This color, which color is this? A lightish beige color with the main outbreaks that have taken place. So we had a main outbreak, a large outbreak in Spain in the year 1613. There was an outbreak in 1735 in North England, an outbreak in 1921 where there were more than 200,000 cases and 15,000 deaths in the US. This led, of course, to a lot of immunization. 1990s, there were outbreaks in Russia, and more recently, there was an outbreak in Indonesia, Venezuela, Haiti, and in Bangladesh. Scientific, why was it called the year of strangulation? When we get to the clinical manifestations, you'll understand why. So I'll remind me to respond to that when we get to clinic. So scientific developments, it was first identified by Edwin Klebs in 1883. So that took almost 270 years to identify the microorganism. And then first cultured and demonstration of exotoxin production a year later. And that was by Frederick Loeffler, who basically came up with one of the important culture media that are used for identification. And then sera and basically vaccination discoveries took place in 1890. Then there was discovery of bacteriophages or corynebacteriophages that carrying the toxin, and basically their role, identification of their role in acquisition of toxigenicity in between 1951 and 1953. And the entire genome sequence was completed in 2003. And in terms of medical development, it was named, or the disease itself was named before the microorganism was identified. So it was named in 1826. That's almost 50 years before the bacterium was identified. Successful treatment with an antitoxin was in 1894. Immunization, 1914. Formulation of the DTP, what is currently used. So the diphtheria, tetanus, pertussis vaccine was 1948. And then assays were developed 1949. WHO included DTP in the recommended childhood vaccines in 1974. And the pentavalent vaccine formulation that, apart from the diphtheria, tetanus, pertussis, including hepatitis B, Haemophilus influenza type B, was in 2006. So a long history spanning very many decades, or is it centuries, that we're still all, I mean, after all these years and the presence of a vaccine, we're still having epidemics being reported, which is quite good. Okay. So as at 2018, most countries actually have more than 90% coverage of the DTP. DTP3 meaning just the original vaccine formulation. So majority of the countries in the light blue have more than 90% coverage. Some of them, there's no data available, probably because it's not being collected. But despite all that, we're having still some countries having large numbers of cases, and this was reported in 2018. So Nigeria having 1,870 cases, Yemen having 2,000 cases, India, despite 90% coverage, still having 8,788. And this, so in 2018, there were 16,611 cases reported, mainly thought to be found or isolated in refugee settlement centers and associated with poor availability of health services, socioeconomic issues, and war may be affecting the vaccination being done properly. So all those means that it's not really being cleared. And once, these are some of the diseases where as long as you have one case, there's still a risk to everyone in the world, being a small village. So how is it transmitted? It's mainly person to person through respiratory droplets, either through coughing or sneezing, but also contact with discharge, mainly from individuals with skin infection or eye discharge, but also through touching of ulcers or open wounds. And we'll see that this is one of the clinical presentation of the disease. In terms of pathogenesis, we have adhesion. So the bacteria has to attach to be able to be internalized into host cells and cause disease. And this is mainly through the presence of pili, which are important for colonization. So it colonizes the nasopharynx, and that may be one of the sources of infection. but there's also adhesion to host pharyngeal cells, and that then leads to internalization. We have adhesins, and we also have hemagglutinins, which I'll not focus on. But the main virulence factor is the toxin. So the diphtheria toxin has two subunits. There's an A subunit and a B subunit. I'm sorry I didn't put the reference for this, but I will. So the B is the binding subunit. It binds to the receptor, gets internalized by endocytosis, and once endocytosed, the B subunit released, the A subunit gets cleaved off, gets transmitted or passes through the transmembrane into the cytosol, and then it ribosylates the elongation factor 2, which then halts or stops protein synthesis. And once you stop protein synthesis, then you're basically killing the cell. So that means that's just a simple way of illustrating how it acts. In terms of clinical manifestations, we have an incubation period of about 2 to 4 days, after which the patient develops pharyngitis or tonsillitis. Within the epithelial cells in this site, there's rapid multiplication of bacteria. It produces the toxigenic strains, so not all of them toxigenic. Please re-explain the mode of action. Okay, so we go back. So someone called student, which you all are, requested for re-explanation. Okay, so the diphtheria toxin has 2 subunits, the active subunits and the binding subunit. So the binding subunit is a receptor on the host cell. Once it binds, then the A and B, both subunits get internalized by receptor-mediated endocytosis. So they bind to the toxin, and that binding to the toxin allows them to be taken up by endocytosis. So they're taken up in this vesicle, and that vesicle then detaches from the cell membrane into the cell, into the cytosol. Now, once in the cytosol, the active subunit detaches from the binding subunit, gets released through the membrane, the vesicle membrane into the cytosol. Once it gets into the cytosol, then it, together with NAD, then it causes ADP ribosylation. That's addition of an ADP ribose to the elongation to the elongation factor. Once that elongation factor gets ribosylated, it stops the synthesis of proteins, and because there's no protein synthesis, then the cell cannot survive, and that kills the cell. So student, are we okay? Not the 268, just the person who is registered as student. I hope you're okay with that. If you still don't understand it, I'll be around on Friday, so we can talk about that. But you read about it between now and then. So as I said, there's an incubation period of about 2 to 4 days, after which there's causation of pharyngitis, that's inflammation of the pharynx and the tonsils and tonsillitis. Within the epithelial cells, the bacteria rapidly multiplies. If the particular strain is non-toxigenic, it probably does not go beyond that. But for those that produce toxins, that's the diphtheria toxin, then they would then produce the toxin locally within the site, so within the pharynx and the tonsils. And that results in necrosis of the tissues and formation of exudate, which causes an inflammatory response. So what then develops in this site is what we call a grayish-white pseudomembrane. So it's a membrane that forms on top of the tonsils initially. So that's the whitish layer that you can see here. It tends to have a texture that's similar to leather and can extend from the tonsils to the larynx and into the trachea. Now, if you then have the pseudomembrane plus accompanying cervical lymph node enlargement, the patient then develops what is described as a bull neck appearance, basically edema of the soft tissues of the neck. So those are the two main characteristics. Now with those two, before I get to that, the other clinical respiratory infections, and this is cutaneous and can range to a small pimple to a non-healing ulcer that can in some cases be quite large. And this is produced mainly by strains that do not produce toxins. So the complications could be either due to the pseudomembrane. If it covers a large area of the upper respiratory tract, then you can have mechanical complication. And that would result in obstruction of respiration and lead to asphyxia. And I hope I answered the question that had been asked about the year of strangulations. Again, it's student, I'm not sure which student, but so I hope I've answered why it was called the year of strangulations, because the pseudomembrane itself was acting as blocking. So the patients were almost like they were being strangulated because they couldn't breathe. Okay, so the other complications are due to the toxins, and this is when it gets into the bloodstream and mainly affects the heart, the kidneys, and the nervous system. With the heart, we have myocarditis, which can lead to congestive heart failure. And then with the nerves, it would be paralysis, and this mainly affects either the soft palate or the eye muscles, leading to complications at those sites. So in terms of lab diagnosis, the disease is not as common nowadays, except in areas where there are epidemics. So most of the time, the diagnosis is clinical. So a good clinical history is very important, always important, at least in medicine, and a good physical examination, because if you don't detect the physical findings, then it's very hard to really ask for the right lab tests. So in terms of specimens, that is always based on where the site of infection is. So the most common would probably be the throat swab, mainly taking a swab from the membrane. In terms of microscopy on gram stain, the gram-positive rods tend to have various shapes and sizes. But as I said, they tend to occur in V or L shapes. That's the palisades. So they are joined together. They do not separate after division. So they'll have either the Chinese letter or the palisade arrangement. And this is, used to be demonstrated in the lab. I'm not sure. I'm sure it will be demonstrated, just not sure how exactly. And then from the specific staining methods, you can actually identify what are known as the metachromatic granules or the volutin granules, and these are energy sources. So it would be this, I'll make it larger at some point when I'm sending it to you, but you can see this roundish structures in the bacilli. Those are what are known as metachromatic granules. For culture, it tends to grow better on media that has blood or serum in it. On blood agar, they usually have a small zone of beta hemolysis. On gram stain, you can see that most of them tend to be joined, they don't separate out, and they have, you can't see the clubbed ends in this, but I had shown you in the previous slide, but they tend to have similar to sort of Chinese writing and that joining. So some of them will have a V-shaped like this one here. Some will have an L shape like this one here. So different, again, different sizes, different shapes will sort of give you an idea of what you're dealing with. On the selective media, which is the potassium tellurite agar, they form blood colonies. And this is, I mean, you see black colonies in the lab that would most likely be corynebacterium. I don't think we have any other black colonies anywhere. So you remember I've mentioned Loeffler as being the person who was first able to culture it. That's the Loeffler's media, and these are the colonies. And for samples cultured on Loeffler's media, or what is known as Dorset egg media, it's much easier to see them large numbers of metachromatic volatility granules. And these granules are only found in toxigenic strains. So those would be the granules within the bacilli. So the granules are the blacks, while the cytoplasm stains gray with the Albert stain. So how do we make other methods of making diagnosis? We have the ELEC test, which is a toxin antitoxin reaction. So you take a filter paper and place or soak it with diphtheria antitoxin, and then at right angles to the filter paper, you inoculate your either control positive and negative, or your test organism. Now, what then happens is, as the antitoxin diffuses into the media, and if you have a toxigenic strain, which also, I mean, the toxin is also diffusing the media, where the two meet, you get what is known as a precipitating line. Just to show you this in an actual culture media. So in this one, which I got from this paper, they have two different ways of carrying out the ELEC test. In the conventional test, this would be similar to what I've just described. So if you have a positive, then you'll have that precipitating line forming. But where you have a negative, so in this one we have positive and a positive, so that's why you have that V kind of shape, because both of them are forming precipitating lines. But where you have a negative, which would be this one, then you only have the precipitating line from the upper inoculum, but not from the lower, so they do not form a V. The modified ELEC test is a different kind of inoculation. So you have the antitoxin in the middle, and then you inoculate the test culture or test microorganism around it, and it's a similar concept. So where the test organism meets with the antitoxin, you have that precipitating line forming, and that tells you that this would be a non-toxygen producing strain, because there's no precipitating line at this point. And this, again, you should be able to see in the lab. The other methods would be culture of tissues and carrying out cytotoxicity tests. Please repeat. And that is from me to me. I wish you had your names because that makes life so much easier. Okay, let's go. Okay, so let's explain this again. So this ELEC test is a reaction between the toxin and the antitoxin. And where the two, or where the reaction takes place, you have precipitation taking place. So you take a filter paper that is soaked with the diphtheria antitoxin, and then at right angles to the paper, you have inoculated your isolate. That's your isolate from culture. So it can either be a toxigenic or non-toxigenic. Now with the non-toxigenic, because it's not producing a toxin, there is no reaction with the antitoxin. But with the toxigenic C. diphtheria strain or toxigenic strain of C. diphtheria, there's production of toxin which diffuses. This you're doing on a culture with media on it. So this toxin that diffuses into the culture media. At the same time, you have the antitoxin that's on the filter paper diffusing into the culture media. Now, where the two meet is where you have formation of a precipitin line. Now, moving that to real life. So if this on your left with a conventional ELEC test, you have, it shows you like what microorganisms are on the plate. So you have a positive one, a test positive one. This is a negative strain. This may be positive or negative. So, and then this is positive, this is positive, and this is negative. So on the actual culture, what do you then see? Since the top inoculum is positive, you see formation, positive for the toxin, you see the formation of the precipitin line. The second one is the test. So we know this is a control positive test. You have formation of a precipitin line. Now, why they are then on the lower and the upper is because the two of them are positive. So it forms a V. This, the third one is a negative. So there's no toxin being produced. So the toxin is only forming from the top one, which was positive, but there's no precipitin line forming from the lower side. And similar to the next space, so there's no toxin because this is negative, there's no toxin from the upper line. T is just a test. It's a positive, it's a positive control. So the positives are the controls, the T are the test, the isolate that you're testing for, whether it's test positive, toxin positive or negative. Are we okay? So the ones that are positives are confirmed positives, the negatives are confirmed negatives, the T are what you're testing. So you've taken a sample from a patient, you want to know whether it's producing toxin or not, so it's the test microorganism. And then for the modified, you have your antitoxin filter paper in the middle and your isolates on the outside. Okay, thanks, SH. So again, here your tests are what you're testing for. These are your positives confirmed, and this is your confirmed negative. So you then have precipitin lines forming between the positives for sure, and none with the negative. And then you make a conclusion for your test organism based on what, whether this precipitin line similar to the positive or lack of a precipitin line similar to your negative control. I hope that is clear now. So you can also carry out cytotoxicity tests. PCR, we said nowadays, is how do you know the precipitation is formed on the diagrams present? How do I know this precipitation? Because of that white line, this white line. And for this one, this circular white line. With the negative, which is at the bottom, you can see there's no completion of that circle, which means there was no precipitation occurring. I hope we're okay with that. If you're not clear, please let me know. So PCR can also be used for detection of the toxin genes. And ELISA tests can also be used for detection of toxins using antibodies for detection of toxins. I thought I'd put this in here, not so much to stress you out, but it's something you may want to look at because it takes you all the way from clinical specimens to which agar that you're going to use. And that's why it's important that you give the lab a complete history, because if I don't know what you're looking for or what you suspect it is, then I may use media that the microorganism does not grow on, and therefore I will give you at the end of the day that there was no growth obtained, while if I knew that you as a clinician have shown or have seen a pseudomembrane, then I know which media to specifically select and then work it out. I'll send you this presentation, so you'll be able to just look at that table. You may use it maybe in your clinical years. Okay, so in terms of treatment, apart from just clearing the microorganism, you also want to neutralize the toxin. So one of the most important treatments is the diphtheria toxin that has been administered, because it's actually the toxin that's going to kill the patient by causing the pseudomembrane and therefore strangulating the patient, as they called it at the first epidemic. But at the same time, you also want to eliminate the pathogen, because if it's toxin producing, if you give the antitoxin but don't clear the pathogen, then they'll continue producing the toxin. So these are some of the antibiotics that it's susceptible to: penicillin, cephalosporins, erythromycin, tetracycline. In terms of prevention, there has immunization. So the diphtheria toxoid, which induces the production of the antitoxin. But however, that does not prevent carriage, and therefore, if you have a carrier within the nasopharynx, they can also transmit it, they can also pass the microorganism to other people, and if they're not immunized, then that can lead to disease. And that's why it's important to vaccinate people, everybody if possible. Okay. So we move on to bacillus. Anyone with a question on corynebacterium or anything that's not clear? Okay, so we move on to bacillus. Again, these are gram-positive rods. They tend to also vary. That's the genus, not specific species, but they tend to vary from a coccobacillary shape to long-chained filaments. They are aerobic. They form spores. So this would be one of the differentiating characteristics from corynebacterium. And the spores are either round or oval. They tend to be in different positions. They are either in a central position or in subterminal midst, close to the end, but not right at the end, or they are at the end of the bacillus. And I'll show you, I think, examples of that. The spores tend to be found in soil. From soil, of course, they can spread to dust, to water, into the air. There are also common contaminants of culture media, and that's why taking care of the environment within the lab is very important, because if you don't, then you'll tell people that they have bacillus while they don't. And the spores tend to be resistant to heat, to drying, that's desiccation, radiation, and to disinfectants. Most strains that are motile tend to have a peritrichous flagella, which I believe you know what that is. And these organisms are catalase positive. So the main pathogen is bacillus anthracis, which causes anthrax. But we also have quite a number of species, though I'll only talk about these first two, mainly about the first one. But I'll mention something about bacillus cereus, which causes opportunistic infections as well as gastroenteritis. In terms of history, origins are thought to be as long ago as 700 BC, but the first clinical description was in 1752. In 1877, Robert Koch, whom I assume you've had a lot of or are going to hear a lot of, used anthrax to develop what are known as the Koch postulates, which I'm sure you've discussed in earlier classes. And the first vaccine was by Louis Pasteur in 1881. The first use of anthrax as a biological weapon was in the First World War in the 1900s. In 1937, the vaccine for animals was used and was found to reduce human cases. 1960s, again, the threat of bioweapons, which I think is still ongoing, was identified. Then there was an outbreak in 1977. In 2001, again, there was an attack in the Americas, and there's a new form of anthrax, which Al mentioned in 2010, that was first described in 2010. That's mainly in people who use illegal drugs using injections. So in terms of epidemiology, between 2005, 2016, there have been large numbers of cases in Canada and some parts of Asia, and that seems to vary. Africa seems to have very few cases, which I don't think is really true. That may be because it's not really being identified, but the eastern part of Africa seems to be doing a relatively good job of identification, not so much, and South Africa, not so much the rest of Africa. But it's thought to cause about 20,000 to 100,000 cases annually, and that's globally. Transmission, there are three main modes of transmission. I think we'll add a fourth one. I should have added a fourth one there. But when you have them in the environment, the spores on the ground can get inhaled, causing inhalational anthrax. From animals, you can have either ingestion of contaminated meat, causing gastrointestinal anthrax, or transmission through the skin, and that is from spores found either in carcasses, in wood, or in hide, and that enters through pre-existing wounds or bricks in the skin, causing cutaneous anthrax. So humans will usually get infected through contact with animals or their animal products. Lab accidents have been known to occur, and this would mainly cause either cutaneous anthrax and maybe inhalational anthrax, or if you touch your culture media on Friday and put it in your mouth, you could also get intestinal anthrax. But then, as you mentioned, there's biological warfare where individuals refine the anthrax spores using specialized techniques, making spores that are very tiny and therefore very easily aerosolized and therefore also easily inhaled and in large spaces. And that's how it's transmitted as a biological weapon. So in terms of pathogenesis, so I said there's cutaneous, where it's transmitted through the skin. And once the spores get into the skin, they'll usually germinate and therefore produce the actual bacteria and will cause local edema and necrotic lesions. And I'll mention how they do that. It's through toxins. Within the gastrointestinal tract, again, there's germination at the site, causing effusion, mucosal edema and necrotic tissue. And the same thing happens within the lungs. So they're then taken up by macrophages at the site of infection. They're taken up by macrophages, they germinate into the bacilli, and then release through the virulence factors, that is the capsules and mainly the capsules and exotoxins. They would then get into the regional lymph nodes, causing lymphadenopathy or lymphadenitis within that site, and then causing disease or various forms of disease, either septicemia or toxemia, can get into the bloodstream and lymphatics, causing meningitis, and within the lungs can cause edema, and all those can result very rapidly to death. So these are the toxins, but I'll describe them in more details in the next slide. So the Bacillus anthracis through plasmids is able to produce a glutamic acid capsule. And the function of most capsules, as we know, is preventing phagocytosis by immune cells. And what has been noticed with most capsules is that infection or antibodies against the capsule, for example, with strep pneumonia, would confer immunity. But in this case of bacillus, that does not seem to induce immunity to disease. The other important virulence factors is the edema toxin and the lithotoxin. So there are three proteins. We have the protective antigen, the edema factor, and the lithofactor. And the edema factor and lithofactor have to combine with the protective antigen to form the toxin. So as they are, when they are factors, they're not able to cause effective downstream effects, but once they combine with the protective antigen to form the toxin, they then act synergistically to cause disease. So the protective antigen, which would be the PA, is the one that binds or allows binding of the edema factor and the lithofactor to cell receptors within host cells. So the two have to, the protective antigen has to bind to the two factors to be able to then allow them to bind onto the host cells to cause their effects. So the edema factor is and has an enzymatic function where it acts as an adenylate cyclase, and this causes increased production of cyclic AMP in the host cells, and that then decreases the efflux of chloride ions and water and results in edema. While the lithofactor, which would be the yellow one, would enter within the endosomes, and within the endosomes then causes cleavage of, I just forgot what MAP is, but I'll find that out. an enzyme, it's a protein kinase that with the lethal factor then disrupts the transduction of the regulatory system, and therefore by causing that activity is able to cause apoptosis, that's killing of the cells, the host cells, but also release of pro-inflammatory cytokines, and that eventually leads to shock. So when you have the factor combined with the protective antigen, we have the edema toxin, which causes edema, and the lethal factor basically causing cell death. So the names of the factors would remind you of what they cause downstream. So in terms of clinical manifestations, we have different forms. We have cutaneous anthrax, which we said is when spores contaminate wounds. And there are different presentations. You can have a papule developing after about 2 to 3 days. And then we get vesicles forming. So you have these small vesicles, usually around the outer part of the wound. And that then develops into a black necrotic central area, which is called an scar. Are the slides moving? Yes, I believe so. Caroline, are your slides moving? So eschar is one of the end stage of cutaneous anthrax, and that is that black portion that you can see on this slide. Then we have inhalation anthrax, which is also previously used to be known as the wool-sorter's disease. It apparently looks like a hole, but it's actually, it's a black layer on top of, yeah, but it does actually look like a hole, but it's a black scab that forms on top of the wound. So that's characteristic of cutaneous anthrax. But of course, there are patients that you will see at these different stages. So it's always good to know that that is like the identification of anthrax, but this could also be. These are also presentations of anthrax, so it's good to know. When you see a patient with this, then you need at what level of suspicion. If you send this, if you have fluid discharging from this and you send it to the lab, then you need to also have asked the patient about contact with animals, especially dead animals or animal products. Right? Okay, so we have inhalational anthrax or wool-sorter's anthrax disease. That's what it's also called. And this is mainly from spores that are inhaled from contaminated hides. Wool lab, please don't open the culture plates on Friday. And then also used in biological warfare. It tends to have two phases of disease. In the earlier phase, about 2 to 5 days, you'll have flu-like symptoms. So the patient presents with sore throat, mild fever, they're tired, and they have muscle aches. Some of them will have hemoptysis, nausea, difficulty in breathing, painful swallowing may be present. But then in the more advanced disease, you have the rapidly progressive respiratory failure. Patients could develop bacteremia, that's the presence of bacteria within the blood. So basically at this point, we've entered into the bloodstream or into the lymphatics. And patients may present with shock, meningitis develops in about 50%. And for most of this, they tend to be almost always fatal. And especially if you're not, if you don't diagnose it and give proper treatment. Now, moving from the first point where they develop respiratory presentation to death usually takes less, about 24 hours. So it's early diagnosis, identification, and treatment. And for respiratory presentation, I think one of the main presentation is that widening of the mediastinum. That space is usually not that large. It's supposed to be much smaller. So it's characteristic of, almost characteristic, definite diagnosis of inhalation anthrax. Then we have gastrointestinal anthrax, but when you're swallowing, of course, you start from the mouth. So you may have oropharyngeal presentation where there's basically neck swelling due to enlarged cervical lymph nodes and edema in the soft tissues. But there's also inoculation into the mucosa, which presents with abdominal pain, nausea and vomiting, anorexia, and they may present with bloody diarrhea. And we said this was discovered in 2010. So injection anthrax, and this is in intravenous drug users. So this could be from two sides. So either contamination of the needles that they're using, or from using intravenous drugs, illegal drugs that are contaminated with the bacillus. So they present with redness and swelling at the injection site. Usually there's no presence of the eschar, so that would differentiate this from cutaneous anthrax, and it may progress to an abscess, and then they also present with fever. So again, this is probably not the complete list of specimens, but it depends on what sample you're dealing with. But they would include wound swabs, vesicular fluid from the wounds or from the initial presentation of cutaneous anthrax. You can have nasal swabs, sputum, blood, and cerebrospinal fluid. And microscopy will show gram-positive rods in chains, and they tend to be attached end to end, forming what is known as bamboo stick appearance, but they do not have spores. Any suggestion why they do not have spores? Though we said it's a spore-forming bacteria. Anyone? When do spores form? Nobody's talking. Okay. So they do not have spores at this point. If you want to know why, find out why, but you can also discuss it on Friday. Absence of nutrients. Thank you, Vivian. So spores will develop in absence of nutrients, but when you're dealing with tissues, there's plenty of nutrients, so they will not form spores. So it provides a conducive environment. And then you can carry out what is known as a McFadden stain or the India ink stain, and that is for the capsule. I won't get a good enough picture online for this, but I believe it should be demonstrated in the lab on Friday. If you don't see it, please let me know. But if you don't see it on Friday, please let me know. Or yeah, whenever you do the practical for bacillus. So culture tends more commonly done on blood agar and nutrient agar, at about 35-7 degrees. The colonies tend to be about 2 to 4 millimeters, which is relatively large. They tend to be white or grayish white. What is unique about them is these irregular margins that are due to the bacteria growing outwards. And these are known as the medusa head. It's characteristic for Bacillus anthracis. So you see this, you're almost definitely sure that it's Bacillus anthracis. And they also have what has been described as comma-shaped projections, which you can see not so clearly, but this is one of them. The colonies tend to be quite sticky on the culture. Don't ask me whether you demonstrate that in the lab, but most of these are supposed to be done in a biosafety cabinet, just to protect. Can you repeat why there are no spores? There are no spores in tissues, but there are spores on culture. Why there are no spores in tissues is because there's plenty of nutrients in the tissues. So once you take, let's say, like the blood sample or your tissue from the cutaneous anthrax, it has plenty of nutrients, plenty of food to feed on. So at that point, it doesn't really need to form spores. In fact, even in cultures, they tend to form spores in older cultures more than in young cultures, because with the young cultures, the media still has plenty of nutrients, but as the culture stays longer, then you'll have spores forming. RO, is that okay? I hope that's clear. Okay, and then the colonies are non-hemolytic for Bacillus anthracis. When you then do gram stain of these colonies, then you get gram-positive rods with a central spore or a subterminal. I don't think we have any that subterminal. So subterminal just means it's closer to the end. So it would be somewhere here, but not right at the end. It's not a terminal spore. It's closer to, it's either central in the middle or away from the middle, but not at the end. And these spores do not extend the cell wall. So they are within the cell wall. I think you'll go through another microorganism that has a spore that extends this, like spreads out the cell wall. So these do not spread out, swell the cell wall. And then you have to identify your colony. So you do your catalase test, you do your motility test, and you do your sugar fermentation test. So they are catalase positive, they are non-motile, and they ferment glucose. Have you ever seen a motility test? Out of curiosity. The non-hemolytic concept. You've never seen a motility test. And the sugar fermentation? The sugar fermentation tests? Yes, I think you saw those on Friday. The catalase test? Yes. Okay, have you done the catalase test? Have any of you done the catalase test? No, it's been demonstrated. You've seen it done. Yes, okay, thank you. So the sugar fermentation, the non-motility test, if it's not there on, okay, I'm not sure what practicals you're doing on Friday, but if you do the bacillus practicals and you don't, the non-motile, the motility test is not demonstrated, kindly let me know. I just forgot. Or alternatively, I could also add in a picture when I'm sending the slides. Just to be on the safe side. Okay, so in terms of treatment, the drugs of choice is ciprofloxacin and doxycycline. And some of the strains of Bacillus anthracis have shown resistance to penicillin through production of beta-lactamase enzyme. So we prefer that penicillin is not used. The other two treatment options have shown variable effects. So the use of anthrax immunoglobulin and monoclonal antibodies. The monoclonal antibodies are basically antibodies produced in the lab from cells or cell lines, and they've been used in the treatment of many other diseases. But these last two have shown variable effects. Some people say that if used in combination with antibiotics, they produce better results. In terms of prevention, we have vaccines, and that's both for humans and animals. For humans, mainly to people at risk, so individuals who work in, who get into contact with animals, for example, veterinary officers or people working in butcheries should have the vaccine. Whether they get it here or not, I'm not sure. And then those also at risk of exposure for inhalational anthrax. So people dealing with carcasses or hides and wool should also get vaccinated. There is also use of antibiotics, not as treatment, but for prophylaxis for individuals who've been exposed, mainly for inhalational anthrax, and where the spores have been released as biological weapons. And then, of course, there's proper disposal of carcasses. If a cow dies, if it dies with really, it should be inspected and then disposed if it's identified as having anthrax, but we know that people eat them, which is not good. And then provision of protective clothing for workers. Laureen, what do you mean if antibodies are what? Lauren Kemonto, if antibodies don't confer immunity, how do monoclonal antibodies work? So by immunity, I mean long-term immunity, whereby if you give an antibody or production of an antibody to a capsule, for example, when the strep pneumonia or the pneumococcal vaccine is it's related to a capsule. So the capsule for strep pneumonia would induce production of antibodies, and that has been shown to give long-term immunity. In the case of bacillus anthracis, the capsule does not provide that long-term immunity. Now, for these monoclonal antibodies, they're not being given for long-term immunity. They are being given for treatment to bind the microorganism and induce an immune response. So it's not long-term immunity, it's just treatment for the disease at the time that it occurs. Is that okay, Lauren? Okay, if it's not clear, kindly let me know. Okay, thank you. So then we have the other organisms that are not bacillus anthracis. So we said we have a capsule in anthracis. The rest of the bacillus species do not have capsules. Where anthracis forms long chains, the rest form short chains. Where it's non-motile, the rest are motile. We said the medusa head colonies are characteristic of bacillus anthracis, the rest do not form that. We said bacillus anthracis is non-hemolytic, the rest cause beta hemolysis, and bacillus anthracis in broth it does not cause turbidity, whereas the rest cause turbidity. Okay, anthracoids are basically all other bacilli that are not Bacillus anthracis. Okay, so something small on Bacillus cereus mainly causes food poisoning, and there are two forms of the food poisoning. We have the diarrheal syndrome, which is mainly driven by an enterotoxin and is associated with ingestion of different types of meats, whether beef, poultry, or other related foods. And in this, we have abdominal pain and diarrhea. Then we have emetic. Emesis basically means vomiting. And the emetic form is associated with ingestion of rice, potatoes, pasta, and dairy products. With the rice, what has been known, it's been associated mainly with Asian foods, mainly, I mean, not Asian food, foods, fried rice mainly made the Asian way. And it's thought to be related to storage of that rice at about below 60 degrees and reheating without allowing adequate cooling, or storage without adequate cooling, and then reheating the same food allows the bacteria to multiply. And this causes abdominal cramps and vomiting. So these are the main presentations, but then we also have other symptoms outside the gastrointestinal tract. This involves the eyes, and this tends to be mainly after trauma to the eye. Then the spores get into the eye and cause endophthalmitis. We also have bacteremia and endocarditis, and this there are many more. I just listed the ones that tend to be more common. In terms of function, there are two types of toxins. We have the heat labile and the heat stable. The function of the heat stable has not been clearly defined, but it's thought to act as a super antigen similar to the one of Staph aureus. I think you did that, where it causes something similar to a cytokine storm. So, but then its actual activity or mode of action in Bacillus cereus has not really been defined. For the heat labile enterotoxin, it seems to have an action similar to that of Vibrio cholera, which you've not done, where it stimulates the enzyme adenylate cyclase, which increases the production of cyclic AMP within the enterocyte, leading to an increased secretion of water and chloride ions and leading to massive watery diarrhea. And the organism has been found to be beta hemolytic on blood agar. I'll not go so much into its diagnosis, but one of them is the beta hemolysis, which is clearing of the area around the colonies. And it also has, sorry, this is not as clear as I thought it would be, but I'll enlarge it before I send it in. You can actually see the spore within that clear area within the bacilli would be the spore. But you can also use serology for diagnosis. You can use toxin detection, and for toxin detection, you can also identify the genes that code for the specific toxins. Treatment is mainly supportive, not so much of antibiotics administration. It's a question. Could the practicals be online? Because for the last two practicals, it was just demonstration. We've not done any hands-on. I don't see the point of coming to the lab. I'll talk about that maybe after this. Just remind me, please. Okay, so in terms of prevention, we have food handling procedures should prevent the multiplication of the microorganism and proper food storage. So again, that should be clear because it's mainly related to how food is stored or how food is handled. Now, then we have the use of bacillus within the laboratory as biological controls. And I'm sure you talked about this when you talked about sterilization and disinfection. So Bacillus stierothermophilus apparently is now known as Geobacillus stierothermophilus, and these are heat-resistant endospores used in the autoclave and other sterilizers. And then we have Bacillus atrophius, which are resistant to ethylene oxide and therefore are used in the ethylene oxide sterilizer and the dry heat sterilizer. And that is all, but before you leave, I just wanted to show you this. I was talking to some of your classmates about summarizing or making life a bit simpler in microbiology. So you're going to have or go through a lot of microorganisms, but it's always good to find an easier way of remembering things. And for me, this is what works, just summarizing things by their groups. So you have gram-positive bacteria. You've already gone through the cocci. And for the cocci, based on catalyst, they can be positive or negative. Positive being Staph aureus. For the staph, you know, they are grape-like. You can differentiate them by coagulase. Coagulase positive, Staph aureus, coagulase negative. You have two species, use novobiocin sensitivity. If they're sensitive, epidermidis. If it's not sensitive or resistant, Staph saprophyticus. And then below this, you can actually write out like what the virulence factors are and what clinical diseases they call. Do the same for strep. Sort of summarizes it quite easily, below which you can have your summary of notes for your revision. Makes life much easier. And for bacilli, you've already done 2 of the aerobics. And for this, you have something like this. And most of them are available online. That's why I didn't remove the name. I shouldn't remove the name anyway. So gram-positive bacilli, you have this you've not done yet. Then you have the non-branching, basically they are rods. So clearly rods, this would be branching rods. And in this, you have the spore formers, where you've already done the bacillus. You have the non-spore formers, which are non-acid fast, and you've done corynebacteria on this list. So it's good to always just have a breakdown, of as you go through. It makes it so much easier to remember. There are 1,000 microorganisms that you're going to deal with. So there's a question here. Why would parasites in the bloodstream rely on anaerobic metabolism? In the bloodstream. So usually the microorganisms that are in the bloodstream will tend to be aerobic or facultative anaerobes. So facultative anaerobes can survive both in the presence or in minimal oxygen content. But they may have a very short phase in the bloodstream and get delivered to dying tissues. And once they get in the dying tissues, then that's where the strict anaerobes, which I don't think you've done any of. I think that's the Clostridia that you haven't done yet. that's where they would survive. Now, there's a question about practicals. And the question is whether they can be online, because it doesn't make sense to come to the lab, right? How many people are in agreement with that before I pass a message on? Please don't send me 270 responses. Okay. My only problem is with somebody who has a microphone on that passed in my eardrums. Disagreed. Some people agree, some people do not agree. Okay. I think I discussed a bit of this with a few of your classmates, and I will discuss it with the lab, whether we can split you into 4 groups and see whether you can actually see the plates yourself, because I can assure you what you see, like what I've shown you on my slides and what you see in real life could be totally different. And considering once you get to the ward rounds, you could actually get sent to the lab to make some of these diagnoses. I would rather that you actually see them. But I'm going to have a discussion with the lab technologist and the chair to see how we can work this out. Kindly give me this week. So turn up to the lab this week, and I'll see how we can work it out. Is that okay? Since guys have started disappearing, I will assume that that will work. But thank you for the lecture. Shall we get the slides for the previous session? The lab sessions are not, the lab slides are not up to me because I'm not the one who does them. But student says the physical practicals remain intact, please. So since student represents all students, I'll take that as what students have all said. Anyway, I will send my lecture slides to your class rep. Please do not sell them. I spent a whole week preparing them. So don't sell them. And if you sell them and make money, please give me something. Anyway, just kidding. So thank you. And if you have any questions, I'll remain for a few minutes.